Okay, we're going to talk about metabolism and switch to my share. Okay, so metabolism as an overview includes both anabolic reactions and catabolic reactions. Anabolism reactions are, combine smaller molecules into larger ones via dehydration synthesis. Endergonic reactions store energy in the bonds that they create. Catabolism is the opposite. This is where we break down larger molecules into smaller molecules. And chemical reactions to break down complex molecules into simpler ones using hydrolysis. Right? Add water, break bonds, release energy. So exergonic reactions are when we release stored energy. Endergonic, putting something within, means we're going to create bonds and store energy in the bonds that are created. In catabolism, most energy is lost as heat when we break things down. The rest of the energy is transferred into ATP. Remember that we looked at ATP in AMP1 also, adenosine triphosphate, that that third, or the phosphate at the end of the 3-phosphate chain, when that is hydrolytically broken, that bond, it releases a lot of energy. If we have adenosine triphosphate and we break the bond and release one of those phosphates, we're left with adenosine diphosphate, ADP. ATP is used for anabolic reactions and for cell work, muscle contraction, any kind of active transport or pumps that need to be maintained. Okay, so every cell needs lots of ATP for maintaining homeostasis, so to speak, within the cell itself. So stage 1 is the digestion in the GI tract, lumen to absorbable or lumen to absorbable forms. Transport is via the blood and the lymphatic system to the tissues and cells. All right, so the first thing we have to do, digestion in the GI tract, of our organic families to the absorbable molecular form. And that's what we looked at in the last, few lectures. We talked about digestion overall, along with the whole digestive alimentary canal, system. We looked at chemical breakdown, enzymatic breakdown of, larger molecules to smaller ones, because it's the building blocks that can get absorbed. All right. So we're going to look at primarily carbohydrates, but it shows you here how proteins, when they get broken down into their amino acids, right, can enter and be used as energy in glycolysis, in the formation of acetyl CoA, and in Krebs cycle less frequently. So we can use proteins, as a molecule to break down and produce ATP. We generally use carbohydrates. Glucose is the preferred molecule, but we also burn fats for energy too, right? So we can break down fats to a glycerol and its fatty acids, right? The glycerol can enter into glycolysis and then produce ATP. Fatty acids will enter in the formation of acetyl CoA, and from there it will reach a certain number of ATP, as these molecules then pass into the Krebs cycle and through what's called the electron transport chain. Right, so first we have to have molecules broken down to the building blocks. Once they're in, they can be used, to along this cellular respiration pathway to produce ATP. And then stage 3, the oxidative breakdown of stage 2 products occurs in the mitochondria of tissue cells. Carbon dioxide is liberated, hydrogen ions, hydrogen atoms, are removed and ultimately delivered to molecular oxygen, forming water. Some energy is released and used here to form ATP. All right, so what does that mean? It means that along this entire pathway, and we go through a lot of processes and chemical reactions to break down one glucose molecule we're going to follow into, reaping the final number of ATP. So what it's saying is that during each phase, so to speak, glycolysis, the formation of acetyl CoA, Krebs cycle, is that we're going to be producing carbon dioxide, which is the carbon dioxide we have to eliminate from the body. And you can see where carbon dioxide is being, a molecule. Actually, it's going to be 2 molecules per molecule of glucose, but you can see carbon dioxide on the blue arrow, peeling off there from the Krebs cycle. And we also produce some carbon dioxide up here, actually in the formation of acetyl CoA. But that we produce carbon dioxide, and that's liberated. We take these hydrogen ions, or hydrogens, and we attach them to some other molecules, which actually are vitamins, and they accumulate and get used right at the bitter end here during what's called oxidative phosphorylation in the electron transport chain. So this is the point, where we reap the most energy. Okay, so carbohydrate catabolism or cellular respiration. All carbs are broken down into glucose, C6H12O6. This is the preferred source or molecule for synthesizing ATP. Remember that insulin is needed for glucose uptake into the cell. So ATP synthesis, there are 2 different ways in which we can make ATP, or the cell makes ATP during this entire process. So the 2 mechanisms are used to make ATP from captured energy that's liberated during cellular respiration. We have what's called substrate level phosphorylation and oxidative level phosphorylation, or phosphorylation, you would say. So substrate for phosphorylation, high energy phosphate groups are directly transferred from phosphorylated substrates to ADP. So what we're saying is you have an ADP molecule. It just needs one more phosphate group added to it. Instead of breaking a bond and releasing energy, we want to create more ATP. So if we have an ADP, adenosine diphosphate, if we take one inorganic phosphate and we synthesize a bond between those 2, then we have ATP. So when we're taking an ADP and we're getting a phosphate group from another molecule, and now we're putting it to the ADP and creating ATP that way, it's called substrate level phosphorylation. It occurs twice in glycolysis and once in the Krebs cycle. Necessary enzymes are in the cytosol for glycolysis and in the mitochondria in the Krebs cycle, or in the mitochondria for the Krebs cycle. All right, so there are certain places where we create ATP along within these 4 phases that the method or the mechanism is called substrate level phosphorylation. And this is something that's really helpful to know when you go into microbiology, because they're going to make a distinction between these 2. Or we have what's called oxidative phosphorylation. More complex process, but produces the most ATP, which means the last phase of this, the electron transport chain, is where we see oxidative phosphorylation. Chemiosmotic process couples movements of substances across a membrane to chemical reactions. Essentially, what's going to happen is that as hydrogens flow through this protein molecule that acts kind of like a turnstile, as it flows down its concentration gradient, it provides the energy, to produce ATP. And that is a very efficient way, of producing ATP. It's why we're going to take all these hydrogens and attach them to NAD and FAD and be used right at the end. Those molecules all become used right at the end. And we move these hydrogens and electrons to this outer membrane of the mitochondria. And when it floods back through this turnstile, it can, it generates energy and is stored in the molecule of adenosine triphosphate. So energy released from oxidation of food is used to pump hydrogen ions across the inner mitochondrial membrane, creating a steep hydrogen ion concentration. And as hydrogen flows back through the ATP synthase, in the membrane, which kind of looks like a turnstile, there's a channel protein, right? So the ATP synthase membrane channel protein. Energy from the flow is used to phosphorylate ADP. right? So there's two different methods. And when we look at each phase, if there's ATP produced, I will mention down there on the bottom via oxidative phosphorylation or via substrate level phosphorylation. Okay, so glucose metabolism or catabolism is cellular respiration. Right, there are 4 steps, so you need to know the 4 steps. We have glycolysis, which occurs in the cytosol. Then we have the formation of acetyl CoA, the Krebs cycle, and the electron transport chain. So the glycolysis, that occurs in the cytosol, and then the end products of glycolysis are going to be shunted into the mitochondria for the last 3 series of reactions or phases: formation of acetyl CoA, Krebs cycle, and electron transport chain. Okay, so glycolysis occurs out in the cytoplasm, and it does not require oxygen. Now, at the end of this, you're going to require oxygen, right, to get into the mitochondria and be able to complete the last 3 phases. But glycolysis, just think of it this way: the phase that's in the cytosol itself, does not require oxygen. So in each of these slides here, I'm going to indicate what you're starting with, and then on the bottom, what you end with. All right. So we start with, on the top, 1 glucose molecule. It's a 6-carbon chain. And what we want to do at the end of this, through a series of some 12 reactions, is that we end up splitting it into, ultimately, 2 3-carbon chains. So I'm going to kind of cut it in half. But the molecule has to be rearranged, multiple times to be able to do that. So in the process, we're going to produce other molecules, like on the bottom, some ATP, the pyruvate, and 2 what's called NADHs. And the NADH is transporting, capturing one of these hydrogens, and it's going to take it down to the electron transport chain. So all this NADH and FADH that we produce along the way is used right at the end. All right, so does not, so that we start with the glucose 6-carbon chain, does not require oxygen. That is anaerobic respiration. It occurs in the cytosol. The NADH transports high energy electrons to the electron transport chain. Boom, that's it. That's all you need to know. We're going to produce NADH. It captures some electron, and it's going to, transport, along with the hydrogen, transport them to the last phase. So you end with 2 3-carbon chains called pyruvic acid or pyruvate. Pyruvic acid conversion depends on oxygen. So if we have oxygen at the end of this process, then this pyruvate is going to go to the next phase within the mitochondria. If we don't have oxygen and we follow an anaerobic pathway, it's going to convert that pyruvic acid into lactic acid. And that gets reconverted to pyruvic acid in the liver, and ultimately, can be used for energy. So if we don't have, even though the reactions within glycolysis themselves don't require oxygen, at the end, you need to have some oxygen. If you don't have it, it gets converted to lactic acid. That's what builds up in your muscles. When your muscles are working really hard, you're not meeting the metabolic requirements of skeletal muscle, providing enough glucose and oxygen, and you start to produce lactic acid. You start to get that burning in your muscles. That does, you can eliminate, so to speak, the buildup of lactic acid by just slowing down the activity. And generally getting to a level of activity doesn't necessarily have to be a complete stop of activity, to then allow your circulatory system and your respiratory system to bring enough oxygen and nutrients to the cells to, for this process to continue aerobically. Right? So you really need to get more oxygen there. And no worries, I mean, even though it's uncomfortable in your muscles, it will be transported, that lactic acid, to the liver and recycled for more use. ATP formed in this matter is called substrate level phosphorylation. Right? So at the end of glycolysis, we have 2 ATP, 2 pyruvate or pyruvic acids, and 2 NADH. We start with glucose, we produce these other things along the way. And the ATP is via substrate level phosphorylation, right? So we can see, this is just an example, you don't have to memorize all these different steps, but we, start with a glucose molecule, we reap certain number of ATP. We actually produce 4 ATP from this one phase of glycolysis with one glucose molecule, but we have to put in 2 molecules of ATP. So we net come out ahead 2 molecules of ATP. We actually produce 4. It costs 2 ATP to make the 4, so we come out ahead 2 ATP. We produce some NADH, and now we have, the 2 pyruvate molecules at the end of this process. All right. So now we go into the next phase, which is the formation of acetyl CoA. Formation of acetyl CoA. So we start with 2 pyruvate, molecules or pyruvic acids or pyruvates. It gets converted to acetic acid before entering the Krebs cycle. So what happens here? We remove a carbon dioxide. We're going to remove one of these carbons. And it's going to take some oxygen with it so that it travels as one molecule. Again, some hydrogens and electrons are going to be attached to an NAD to produce NADH. Thus, it's going to travel to the electron transport chain to the last phase. Acetic acid can be attached to what's called a coenzyme A or a CoA for transport, and thus becomes acetyl CoA. Right, so we start with 2 pyruvate acids that we ended with in glycolysis. We're now going, remember, this is a 3-carbon chain pyruvate. We're going to remove one of the carbons and some oxygen. That's one of the carbon dioxides that you're producing. And you're going to be left with a 2-carbon chain. We're going to remove hydrogen and an electron with it, because hydrogen has an electron. 1 electron's outer valence shell. It will bind with acetyl CoA or coenzyme A to form acetyl CoA. And now that molecule, acetyl CoA, is going to be the molecule that enters the next phase, which is the Krebs cycle. All right, so we start with 2 pyruvate. We end with 2 carbon dioxides. Right? We have 1 carbon dioxide for each acid. Right, 2 carbon dioxide, 2 NADH, and 2 acetyl CoA. Right, then we enter into the Krebs cycle, and it's a cycle because we go around and around and around. And there's a little mnemonic that I can give you later that helps you remember the intermediate products of Krebs cycle, which is another very good thing to commit to, or process to commit to memory before you get to microbiology. Okay, so now the end products of the formation of acetyl CoA, which are 2 acetyl CoA, are now going to go into and get pumped into this next cycle. Okay. So the 2 acetyl CoA joins with a chemical to form a compound of a 6-carbon compound as it enters into the Krebs cycle. So because there are 2 acetyl CoA molecules, right, that are produced from each glucose molecule, because we started with 1 glucose molecule, now we have 2 acetyl CoAs. You're going to have 2 turns or cycles around this, or passes around this cycle, full cycles for each acetyl CoA. So 1 acetyl CoA enters, and we go through all these different steps all the way around the cycle, and then the other acetyl CoA enters, and you go around another complete turn of the cycle. Called the cycle because the beginning molecule, oxaloacetate or oxaloacetic acid, is generated by the end of the cycle. So at the beginning of the Krebs cycle, we begin with 2 acetyl CoA. Right? It produces ultimately, through its 2 cranks around, 4 carbon dioxide, more carbon dioxide that has to be transported to the lungs, 4 carbon dioxide, 2 ATP via substrate phosphorylation, 6 NADH, and 2 FADH2s. All right, so we start with 2 acetyl CoA, we end with what we produce here. So here is a mnemonic to keep in mind, in succession. the different intermediate products of the Krebs cycle. So as acetyl CoA enters into the Krebs cycle, it actually is going to bind with the bottom, the last molecule here, oxaloacetate or acetyl, I can't say, oxaloacetate, it's easier to say. And when that 4-carbon chain of oxaloacetate merges and combines with your acetyl CoA, it forms the first 6-carbon molecule called citrate. So that's why we start with citrate, because that's your 6-carbon chain. So the first product is citrate, isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, and then back to oxaloacetate. So a handy little mnemonic to keep them in order is, can I keep selling sex for money, officer? I saw that in the internet someplace, but it really works. Right, so you keep repeating that to yourself, and then you do have to know the intermediate products, but it helps you keep them in order, right? Citrate, isocitrate, alpha ketoglutarate, and so forth. So let's kind of take a look at this reaction. All right, so from the cytosol up at the top is showing you, hey, this is where glycolysis is occurring. We end with the pyruvic acids or the pyruvates. Right? You have some oxygen available, right? Because it's kind of that oxygen is going to grab onto that carbon here, right, to form carbon dioxide. All right, so now we enter into the mitochondria, right, and we produce our acetyl CoA. So we start with the pyruvate, which is a 3-carbon chain on top with 3 black balls. All right, we remove a carbon and attaches the oxygen, forming carbon dioxide. Bam. Right? We produce NADH, and we add CoA to form acetyl CoA. It's a 2-carbon chain, 2 black balls with the CoA, not the blue ball. Now that enters the Krebs cycle, right? That's all the green arrows. So we have a 2-carbon molecule entering this product, this process, and it binds with, to the left, your oxaloacetate, a 4-carbon chain. Thus, they merge and form citric acid, a 6-carbon chain. Tells you it's the initial reaction. From there, we kind of rearrange it into isocitrate. From isocitrate to alpha ketoglutarate, we remove more carbon and form carbon dioxide, and we produce more NADH. Then from alpha ketoglutarate to succinyl CoA, we're going to add another CoA. We're going to remove another carbon to form carbon dioxide. We're going to take out a hydrogen and its electron to form more NADH. And now we have succinyl CoA. And look, we're back down to a 4-carbon chain with a CoA. All right, now we're going to go from succinyl CoA to succinate. We're going to release that CoA, and we're going to produce the ATP, 1 ATP. Now from succinate to fumarate, we release another hydrogen or electron and carry it in the form of FADH. Then from fumarate to malate is another rearrangement type of a reaction. And then from malic acid to oxaloacetate, we liberate another or release another hydrogen ion and its electron to produce another NADH. And now we're back to oxaloacetate. Most biochemistry, if you ever take a biochemistry class, is all about these different reactions and how proteins enter in, how fats enter in. You draw generally, because organic chemistry is usually a prerequisite for this, is that you draw out all your molecules. It's crazy chemistry, but it actually makes a lot of sense, because when you draw them out and you make these different ball and stick models and things like that, is that it really helps you really understand what's being added to this, what's being taken away, how is it rearranged, generally in more detail than you will ever need in your life. But hey, these are the hoops that you have to jump through to get into certain programs. So the nuts and bolts that you have to know for the Krebs cycle are what you start with, acetyl CoA, what it produces, right, down at the bottom, I would work on, oops, went the wrong way, I'm sorry, memorizing the intermediate molecules using that handy dandy mnemonic I gave you. But if you can kind of look and see what's happening here, again, for each molecule of acetyl CoA, which you produce now 2 from that 1 glucose molecule, 2 turns around the Krebs cycle. Okay, and then we move on to the electron transport chain. So the NAD and the FADHs are that we've seen that we're putting hydrogens and electrons to and shuttling down to the end of this process to the electron transport chain. So that they are coenzymes that transport electrons and hydrogen from each of the 3 previous reactions. And so we generally, and it's not always exactly the same number, but it depends on what substrates you're using, for energy, whether it's protons and proteins or carbohydrates or fats. But for all intents and purposes, at the end of this process, at the electron transport chain, we should have about 10 ADH, 2 FADH2 that are transporting these hydrogens and electrons. Oxygen is required for this step to occur as well, because we're going to have to, we'll see in just a minute, we're going to take those hydrogens and add it to it and form water. ATP is synthesized as electrons flow along the chain, what's called chemiosmosis. And at the end of the chain, oxygen picks up the electrons, called the final electron acceptor. The hydrogen ions and depleted electrons bind with oxygen to form water. ATP is produced in this manner called oxidative phosphorylation. Ding, ding, ding. And it produces, depending on how a question is asked, but somewhere around, net around 30 to 32 ATP and multiple water molecules. Some questions will ask you, what was the net production of ATP? What was the gross production of ATP? What was the net production of ATP or the gross production of ATP in glycolysis? What about the formation of acetyl CoA? What about in the Krebs cycle? You really have to look at the question. My questions are going to be very straightforward. Right? I just want you to know basically what you're starting with, what you're ending with in each of these phases, and where they occur, what kind of phosphorylation are we talking about? All right, so here they're showing you a picture of essentially what's happening. And up here in the upper left-hand corner, it has a little block around the area it's going to magnify, because here's your mitochondria. Remember when we looked at the mitochondria, we said that, actually in AMP1, that it has an outer membrane and what's called cristae in the center. And it's like another layer of phospholipid. And so you have the outer membrane and then the inner membrane. And this shunting of hydrogens and electrons across the membrane, we're talking about remaining within the mitochondria itself, but not to the outside, within the inner membrane. So when they magnify it, the outer plasma membrane of the mitochondria is to the outside, kind of with the red exterior, and then that inner cristae layer is down below. So all they're trying to show you is that there are different molecules associated with this process. And what's happening is the NADH and the FADH2s, come by or have to interact with this enzyme here in the inner membrane layer. And it's going to essentially take the hydrogens and electrons off and pump it between the 2 membrane layers. They accumulate down here over on the right-hand side where you have phase 2. It says chemiosmosis uses the proton gradient to synthesize ATP. So the proton is the hydrogen, not the hydrogen ion. So you see all the positive charges accumulated, on the superior surface of that membrane where it has all the hydrogens. And now they're going to pass through this ATP synthase. Right? So to synthase something means it's going to produce and put together and form ATP. So as it passes through, it kind of looks like a turnstile. It's going to spin these sides of it, and it's going to bind an inorganic phosphate to ADP to produce ATP on the bottom line. And we produce a lot of it that way, a lot of ATP that way. Right. Right. This shows you kind of the shape, the diagram of the shape of it. You see what I mean? If the hydrogens are moving down their concentration gradient and diffusing, right? They get passed through this turnstile, and when they do, it spins it, and that generates the energy to bind a phosphate to ADP and ultimately, boom, produce ATP. Okay, so here we're talking about the different phases: glycolysis, formation of acetyl CoA, Krebs cycle, electron transport chain. And it's talking about generally, how many ATP are produced or net along this whole process. And it's just showing you here glycolysis, it nets 2 ATP by substrate phosphorylation, and then in the Krebs cycle, 2 ATP, by a substrate phosphorylation. And then over to the right-hand side, about 28 ATP by oxidative phosphorylation. Thus, the total can vary, anywhere from like 32 to 30 to 32, give or take, around in there. There is some variation to it. All right. And it's showing you here between the electron transport chain on the right-hand side to the amount of ATP. It's showing like a little circle there, 10 NADH plus an H plus times about 2.5 ATP are produced, for 10 NADH molecules, and 2 FADH2 produce about, 1.5 ATP. So you just multiply them out, but there's a little bit of variation, just for people who want to know. Okay, we're going to talk a little bit more about how we transport some lipids and things, and what are the difference of these molecules. Lipid transport, remember, it's a non-polar, polar covalently bonded molecule, so it's insoluble with water, must combine with some proteins on the surface of it, to be transported as a lipoprotein. Types of lipoproteins depend on density. The smallest ones are the chylomicrons, and those are actually formed in the smooth endoplasmic reticulum of the simple columnar tissue that lines the mucosa of the small intestine. And then they become VLDLs, LDLs, and HDLs. Let me just back up quickly, back to cellular respiration here. As in case I forget to mention it at the end, is that on the course page in the exam 2 content, below where this, the notes are, for this chapter or for this portion of metabolism, below that is a review sheet. And it's a series of questions. It doesn't give you the answers necessarily, but I think I list about 12 to 15 questions, maybe, that you can answer from the notes and what I've just described to you. And if you write all those out and answer all those questions, and of course, study them, memorize them, the answers, then you will be able to answer the questions that are on the exam, okay, that from that material. So I would fill those out, because I know, there's a lot to cellular respiration, but if you answer those questions and you look up the answers and then you memorize those, that'll answer, I think, virtually all the questions I ask. So just study it. Okay, so lipoproteins, talking about fats. Chylomicrons formed in the small intestine, those smooth endoplasmic reticulum of the columnar cells in the mucosal layer of the small intestine. We said it takes up these fat molecules on its surface like it does other molecules, but now the smooth endoplasmic reticulum becomes really active, and forms chylomicrons. So now it can be, so it's endocytotically kind of taken from, taken in from the surface of the cell, travels to the SCR, and then now is in another vesicle. It is exocytotically released on the basal surface, so it can enter into a lacteal. And then it gets transported through the lymphatic system, through the subclavian veins, and ultimately transported back to the liver. Very low density lipoproteins, or VLDLs, are now the next step, formed by hepatocytes in the liver. They transport triglycerides formed in the liver to adipose tissue, where we store fat. Adipose cells can kind of shrink or swell depending on, the availability of different VLDLs. All right, then we have low density lipoproteins, or LDLs, and LDLs and HDLs are what you look at a lot to identify or to evaluate the general health of an individual, their fat ratios, how many healthy fats versus unhealthy fats that they have. And this is why. Okay, so the LDLs carry about 75% of the body's cholesterol, all right, transported to all body cells. And at high levels, right, they can deposit cholesterol into smooth muscles of arteries. So that's why the LDL, percentage of a blood evaluation, blood chemistry, is important. We want to let that level to be, below a certain range and within a normal range. If it goes too high, it has a tendency to deposit fats into smooth muscle and into arteries. That's not good. However, high density lipoproteins help remove, they're the good ones, they help remove excess cholesterol from cells in the blood and transport it back to the liver to be eliminated. So that's what you want. You want low density lipoproteins, so your body's cholesterol levels are lower, and the HDLs help bind with it and take it to the liver and help you remove what's there. So it's normal to have a certain amount. You just want to have lots of HDLs to capture, the excesses and help it be eliminated from the body. So what we're saying is that when previously where it says the type of lipoprotein depends on density. The less protein you have, the less dense it is, like fat floats, all right? The more protein you put on, the more the ratio goes from being non-polar covalently bonded molecules to polar proteins within it. It becomes denser and denser and denser, all right? So the chylomicrons have the least density. Then you have the very low density lipoproteins, the low density lipoproteins, and the high density lipoproteins, because there's a lot of protein there compared to an LDL or a VLDL. All right, so HDLs are the good ones. Right, lipid catabolism breaks down. This is how we break down triglycerides. Remember, we start with the macromolecule of a triglyceride, and it gets broken down to 1 glycerol and 3 fatty acids. For all intents and purposes, it's not exactly like that. Right? And we use that, we use lipase from the pancreas primarily to do that. Right, the fatty acid chains can get broken down into smaller units, and it does that through beta oxidation. Beta oxidation. Right, so why do we care about that? Beta oxidation and fatty acid catabolism used to make acetyl CoA. So when we are burnt, oopsie doodle, I just got past the wrong one. I'm sorry. I just zipped back to the beginning. All right. When we are breaking down fats, and we will often do that, when we don't have enough carbohydrate to burn. So when we do that and we're burning fats, right, that some of the end products of breaking down these fatty acids during beta oxidation can be used in the Krebs cycle. So when we looked at the Krebs cycle right at the beginning, so you could see where some proteins come in and where the fats would come in, either into the formation of acetyl CoA, or maybe into the Krebs cycle, or maybe just in glycolysis. Right? Well, this is how we can use some of the fats, to make energy and form ATP. So it's used to make acetyl CoA, which enters the Krebs cycle, also produces ketone bodies, which two acetyl groups combined. All right, so a ketone body, right? Ketones, when they build up too high, right, become acids, and acids liberate hydrogen ions, and hydrogen ions decrease the pH of blood. So when we burn lots of fats, higher than normal, not just because you're burning fat because you're running on the treadmill or you're out riding, doing a 100-mile bike ride or something. We're talking about over time. Is that when we use beta oxidation, we have high levels of ketone bodies that are produced called ketosis. And they become acids, and it causes, makes the pH, the hydrogen ion concentration increase because all acids dissociate in water, and the pH to decrease. So it can lead to acidosis, called ketoacidosis. This is common to get to this kind of dramatic level in diabetes mellitus patients, especially diabetes mellitus 1 patients that are not being properly managed. Okay, lipid anabolism, how we build it up. Well, we can take glucose, in different substrates that are in excess, in our body that we ate that we did not store as glycogen, or maybe, in skeletal muscle, either in the liver or in skeletal muscle. So the glucoses can come together to form glycerols and ultimately produce triglycerides. So what this means is that if you eat more carbohydrates than you burn off every day, it gets turned into fat. All food, if you eat excess, if you take in more calories than you burn off, it gets stored. And if you keep doing that, then it becomes stored as fat. It's stimulated by insulin, and occurs in liver and adipose cells. Right, protein catabolism, breaking it down. Well, we know we take proteins, we break it down into dipeptides and ultimately amino acids. Amino acids can enter into the glycolysis, into the Krebs cycle, or as acetyl CoA. So it has a couple different paths. But it must be what's called deaminated. We need to get rid of the blue balls on the protein molecule, right? The nitrogen. So deamination means we're going to lose that NH2 molecule, and it happens in the liver. We'll often combine it with another hydrogen ion, and that forms ammonia. And then as it passes through the blood into the kidneys, it becomes urea and is generally excreted in urine. We never reabsorb ammonia in the kidneys. So in the process of breaking down proteins, it has more opportunity, more phases it can jump into and be utilized and burned. But it does need to be deaminated by the liver and remove that nitrogen group. And that is a toxin, something we're going to eliminate in urine. That's where the urea is coming from. Protein anabolism, building them up, is just in the reverse. We have amino acids that form dipeptides and then longer chains to polypeptides. Remember that this occurs with the ribosomes. Amino acid assembly. Remember the ribosomes? Ribosomes, which perform amino acid assembly in cells. Liver produces most of the plasma proteins. Albumin, fibrinogen, all these different, proteins, anti-A, anti-Bs, if you need those, well, depending on your blood type, of course. Metabolic regulation, right? So we have these two different phases, and now we're about done, folks. We have what's called the absorptive phase and the post-absorptive phase. Right? So the absorptive phase is about the first 4 hours after you're ingesting food. So nutrients entering the bloodstream after a meal pump up, increase your blood glucose levels. Generally within 4 hours of ingestion, right? The dominant hormone is going to be insulin. Shocker, right? And once cell stores glucose, the extra glucose will be converted to fat. All right, so we don't want to be taking in a lot of excess calories. This is why also it's sometimes there's the theory that it's better to have smaller meals during the day instead of a couple very large ones. So once the cells, in skeletal muscle and all your muscles, all your body cells really, and the liver, once they are finished storing the molecule for energy use at a later date, it becomes converted to fat. So we have what's called lipogenesis, the creation of fat. And then the overall effect will be to decrease your blood glucose levels. Insulin, higher than normal blood glucose levels. Store it all, stimulate the conversion to fat, decrease blood glucose levels. But then we have the post-absorptive phase. Now the opposite happens. Generally, somewhere around 4, even maybe 6 hours after a meal, when absorption is complete, or you're entering kind of a fasting state, which means now the cells have used up all the glucose, that they've had available to them. And now your blood glucose levels are dropping lower than normal. Right? So the dominant hormone here is going to be glucagon and some cortisol, a little bit of cortisol, because you remember that cortisol can have an elevating blood glucose effect, but it's primarily going to be glucagon. The overall effect is to maintain or increase blood glucose levels back to a normal range, maintain an appropriate glucose level. Okay, so that is it. That is the end of metabolism.